Okay, welcome to chapter 16, folks. We're going to look at just a little bit about the structure of the nucleus, DNA structure, and some stuff about the chromosomes. And I'm giving this a very, very light treatment because it's really in the realm of genetics. But there are a couple of things that it's good to have a little bit of understanding of if you've not taken genetics yet. So let's get started. First thing we're going to look at here is a little bit about DNA structure. And so you'll be familiar with what is known as B DNA. And so you're familiar with your double helix that you've seen in general biology and in many other biology classes probably for many years. And it's what you've always looked at there. It's called B DNA, but there are two structural variants of DNA, an A variant and a Z variant. So A DNA is a right-handed helix. It's shorter and thicker than B DNA, as you can see in the diagram. It doesn't occur naturally. If it does, there's very, very little of this occurring naturally in cells. And most of the DNA is in the B form. You can also create a Z form as well, and you can see that's got a narrower diameter than both A and B DNA. So it's just kind of good to know that there are some variations of the double helix. If you ever find an RNA double helix that can arise due to secondary structures in RNA or hybridization between RNA and RNA targeting molecules, they're often of the A type though, so those can arise in cells when you're looking at RNA helices. DNA in cells isn't just you know that it's associated with proteins and arranged into chromatin and all that kind of stuff, and we'll talk about that later. But DNA can also have a structure due to coiling. So the DNA double helix can twist upon itself to form what is called supercoiled DNA. Twisted DNA that is twisted even further in the same direction is called a positive supercoil, and if you twist DNA in the opposite direction that's already coiled, it's called a negative supercoil. You find this both in linear and circular DNA molecules, so you find it both in eukaryotic and prokaryotic chromosomes. But it's best and most easily studied in circular DNA. If you completely untwist a DNA molecule and relax it entirely, it's called relaxed DNA. And DNA can go back and forth between negative supercoiling, positive supercoiling, and being entirely relaxed. If you have extensive supercoiling, that can make chromosomal DNA more compact and contribute to DNA packaging. It can also restrict the access of DNA to proteins involved in gene expression. So the way that you relax or create these supercoils is by inducing, is by using enzymes which can change the supercoil, the extent of supercoiling, and that's also known as changing the linking number. And so one enzyme that can do this is called type 1 topoisomerase, or topoisomerase 1, and that will make single-stranded breaks, and it will pass the other strand through the single-stranded break, rotating the DNA helix as it does so, and then resealing the break, and so you can add and relax or add or remove supercoils using that. There are also type 2 isomerase as well, and these induce double-stranded breaks. And if you've ever heard or if you recall about DNA gyrase can do this as well. So two ways of relaxing or creating supercoils: either introducing a single-stranded break and passing the other strand of DNA through, and then rotating the helix to reduce the supercoiling or increasing the supercoiling, depending on which way you do it. And then you can use type 2 topoisomerases, which break both strands of the double helix. Last thing to talk about in this short section is denaturation and renaturation. And the only reason that I'm going to cover this here is because the technique we're going to talk about in this section is called fluorescent in situ hybridization, and it's based on the denaturation and renaturation of double-stranded DNA. So understanding the biophysics of that can help you understand how that technique works. Like you know, DNA strands are held together by relatively weak non-covalent bonds, hydrogen bonds, and strand separation, which is also known as DNA denaturation or melting, can be induced experimentally by either raising the temperature or the pH. And you can then monitor this melting from double-stranded to single-stranded by looking at the absorption at 216 nanometers. So all DNA absorbs light with a maximum around 260 nanometers, so that's down towards the UV range. Might even be in the UV range, I don't know off the top of my head. What happens is that as you increase the temperature and the strands start to separate, we see an increase in relative absorption at 260 nanometers. So if we set double-stranded DNA at a relative absorbance of 1, and then we start to melt the DNA, we see a rapid increase in the absorption of UV. The temperature at which half of the absorptions change is reached, so if that's the maximal absorption at 260, and then half of it would be down here, we call that the DNA melting temperature, or the TM, and that depends on the AT, the adenosine to thymine content, which is obviously then dependent on the GC content. We can then renature or reform the double helix by then again cooling over a period of time, and what happens then is that the DNA will find sequences that are similar, and they will snap back together, and hydrogen bonds will form as we reduce the temperature and wait over a time course. So this is the whole basis of nucleic acid hybridization experiments. You can identify DNA sequences by looking at the renaturation of a piece of DNA that we call a probe, which has some similarity with a target DNA molecule, and depending on their similarity, they will match with at a certain temperature. That's their melting temperature. 50% of them will be re-annealed. This is the basis of techniques such as polymerase chain reaction, but it's also important in this technique called FISH, and it's used even to study aspects of evolution when you're looking at changes in nucleotide substitution, and you can have a probe which will match at certain temperatures given perfect matching, and then it won't match at that temperature if there are some differences between the two, and so you can actually use very subtle differences in the melting temperature to get an idea of the number of differences in two sequences of DNA, and that can give you an idea about homology and nucleotide identity when you're trying to compare DNA from two species, and then you can start to make some inferences about the phylogenetic relationship between those organisms. So we'll finish it there. We're going to come back to this renaturing and denaturing of DNA when we look at this technique at the bottom here, fluorescent in situ hybridization. That's really the only reason I want you to know about it. So I'll stop there. I'll be back soon. We're going to look a little bit about DNA packaging. It's all about histones and chromatin. I'll see you soon for that.